Hi, Rolson. Sorry I was a little late. I had to get my computer out and had to boot it up. I think I accidentally shut it down. It took a few minutes, and I was kind of late on top of that. So, for some of you guys, I'm covering for Dr. McCain today. I think this is usually her slot time, so I apologize if you were expecting her. But let's get down to it. I've wasted enough of everybody's time. Um, we're going to be doing muscle contraction today. All right, muscle contraction is the big one um, that we spoke about. If you were attended one of mine um, lectures last week, then we kind of talked about the neurons and electrical conduction, and I kind of told you about neuromuscular junction, and we left the muscular part up until the following week. So that's what we're going to pick it up when we talk about what happens when the electrical conduction moves into the muscle. And I think next week we're going to be doing this again also. So we'll probably do part two of muscle contraction. We'll go in a little bit deeper on that. Okay, is there any questions, by the way, before I get going? I know perhaps we're on the verge of spring break, which is exciting, and hopefully we'll be able to, push through and get all of our tests done. I won't ramble anymore. Any questions or any topics that we want to talk about outside of muscle contraction? Okay. All right. Okay. So we'll go. We'll just go. We'll dive in. So when the action potential decides to jump in from the terminal baton from the neuron, and it will enter the muscle, it will kind of be dispersed throughout the muscle. Okay. Now, before I should probably go into the mechanism, I should probably tell you what muscles are made of. Okay. Muscles are made of sarcomeres, and sarcomeres are pretty much the building block or the building unit of a muscle. And what they are made of are two different filaments. They're made up of actin and they're made up of myosin, right? We probably remember myosin from mitosis being the train tracks that kind of pull apart the sister chromatids, and actin was more like I think it's more like an intermediate or microfilament. So what it is basically we have this sarcomere that is actin over here and myosin over here. And as we will create tension in our muscles or contraction, these kind of come together and slide towards each other. All right. This is called the sliding filament theory. This is kind of pretty much the sliding filament theory is the idea of what we have of how muscles contract. Okay. And there's things that go on that cause these muscles to contract. Okay, we'll talk about that, and that kind of what happens when the action potential comes into play. So let me make sure I touched on all that I want to talk about. Okay, there's one part. So have we ever? I want to demonstrate this. Has there ever been a time when we want to where we've done a bicep curl and we've noticed that once we do a bicep curl like this, we curl it up and then eventually we see like this bulge. Popeye had this in the cartoons, or we see it here. Not as much as myself anymore. Back in my college days when I lifted weights, but I think you guys get the point where we start to see muscle mass and more like a bulge into our bicep. Well, all that is just the sarcomeres that are contracting, right? And there's so many of them that the actual muscle does contract, and then it produces this bulge in your bicep. Same with your quad, same with your calf sometimes, or your cannot think of your pectorals. That's what it is. Your pectorals also do this too. So it's kind of interesting. That's kind of all that is just all the sarcomeres, probably thousands or a couple 100,000 sarcomeres that make up your muscle, doing all that at once, and this is what kind of creates the tension in the muscle, during contraction. So we've spent a lot, for the short time that we've spoken, we kind of talked more about contraction, right? Well, there was also, our muscles have to relaxation too. and we'll talk about that. So the main player that causes all this, we talked about sodium and potassium. The main player that causes muscle contraction and muscle relaxation is calcium. Okay, we haven't talked about calcium much when it comes to physiology. It's important in the cardiac heartbeat and skeletal muscle, which is kind of what we're going to talk about right now and introduce the topic. Is that it is important in contraction of the skeletal muscle. So when calcium is stored in the sarcoplasmic reticulum, okay, which is inside these muscles. We have the muscle sarcomere, and then we have the T-tubule, which is on the outside of the sarcomere. And the T-tubule pretty much is a network of, let's say, a expanding network that helps kind of propagate the calcium out towards the muscle. All right. So when we have the electrical conduction come in, I might have mentioned that calcium is stored in the sarcoplasmic reticulum. If I forgot to, I apologize. Calcium is stored in the sarcoplasmic reticulum. When electrical conduction reaches the muscle, it will open the channels of the sarcoplasmic reticulum. Calcium will move out, and then the T-tubules will help distribute calcium as much as possible into the sarcomeres. Now we have calcium kind of somewhere spread out into the muscle. We have to talk about how myosin binds to actin. Okay, myosin binds to actin in a way that I should probably say that myosin right now, we have myosin alpha and beta tubulin. Okay, that's what makes up the myosin strand. Along this myosin strand are these, I'm going to call them myosin heads. They're like these little protruding proteins that we call myosin heads. If this is the myosin, what's sticking up is these myosin heads, and they have two conformational changes. And what that means is they switch from different angles. As I use my finger here, more like a cocked position or a trigger position versus more like an inactive. And the cocked or triggered position, all that is just it allows the myosin to bind to the actin better. Now, where does it bind in actin? And why does it not bind all the time if we have calcium? Well, and now we're switching to the actin side of the sarcomere that it's going to bind to. Actin is this filament, and something too exciting about actin, but we have binding sites on the actin. However, when calcium is not around, we have this other molecule called tropomyosin. I don't want you to get confused between myosin and tropomyosin. This is tropomyosin that it somewhat corkscrews around the actin. And it just so happens that when our calcium is not around, this tropomyosin will block the binding sites for the myosin head. Okay, so if we're blocking the myosin head, then obviously we are in relaxation. All right. And then when calcium becomes introduced into the T-tubules and then kind of gets dispersed into the muscle, it will bind the tropomyosin. It'll bind to tropomyosin and cause this tropomyosin to do a shift. Okay. How about now? When's the last time you heard something, Christina?
I can hear now. Just the next, it was like a minute.
Okay. I think my mic got turned off accidentally. What was the last thing we talked about? The
I'm not
acting
sure.
and sometimes this mic turns off on this Blackboard Collaborate. Do we remember the last time we heard something? Okay. Muscle. Well, the last part I kind of talked about maybe was when calcium binds to the tropomyosin. The tropomyosin will switch over the actin or kind of twist on the actin, and that reveals the troponin binding site to the myosin head. All right, and this is kind of how we get muscle contraction. Okay, so just to say that again, when I'm going to take it from the top, when the action potential goes into the muscle, the action potential will open up the sarcoplasmic reticulum to release calcium into the T tubules, which is kind of like this. Think of it as like a highway network that will help distribute calcium throughout the whole um, muscle. And when calcium binds to the tropomyosin, it will corkscrew and reveal the binding site on actin for the myosin head, and that's kind of how we get contraction of the muscle. So I hope you guys kind of all heard that. That's kind of like a synopsis of the last 15 minutes, what I was talking about. Okay, so let's talk about the myosin head because I kind of talked about conformational changes there and what happens when we bind and we don't, when it's cocked and when it's uncocked. On the myosin head, there is adenosine triphosphate, ATP and ADP. Okay. When the myosin head is triggered and ready and it's kind of in this cocked conformational change, it will bind to it is able to bind to the troponin on the actin. So we have to have ADP bound to that. That's how we get contraction on the myosin head. Once it binds, it holds on to ADP, but for it to release, it has to have ATP. All right, it will convert from adenosine diphosphate to adenosine triphosphate. And this conversion from ADP to ATP will uncock the myosin head, and then it will subsequently be released once calcium is pumped back into the sarcoplasmic reticulum. And this is what relaxation is. So I got a good example on this. Do we all know what rigor mortis is? Rigor mortis is a post-death phenomenon that kind of comes on a few days after um, any kind of organism passes away, any mammal with muscles. We've probably all been on the interstate and we've seen some kind of roadkill on the side of the road that appears distended or stiff, or its legs are sticking straight up. We've probably seen it on cartoons. And what this really is it's rigor mortis, and it all has to do with the way muscles contract. Okay, and if you remember from metabolism, or perhaps when we have glycolysis, citric acid cycle, and oxidative phosphorylation, you may remember that we're all just kind of pulling a bunch of electrons off of glucose, right? And we have all these different electron carriers and these electron acceptors because we have to do something with these electrons, right? We can't just pull them off and have nothing. We have to use them. We have to, harness their energy. And what we do is we kind of pull these off and place these on the electron carriers. And when we reach oxidative phosphorylation, what really happens is that at the bottom of the oxidative phosphorylation chain, the final electron acceptor is oxygen, right? Diatomic oxygen, atmospheric oxygen, O2. And when we pop that electron on O2, we make water. Water is kind of like a metabolic byproduct of metabolism. So the point I'm trying to make here is that we're breathing. right? And we're making ATP. So if we are breathing, we have oxygen to accept that, to be that final electron carrier molecule, and then that obviously will make ATP. If you remember, that's what metabolism is. It's energy to make. It's the tools we need to make ATP, right? That is the energy currency of the cell. So it puts that extra phosphate on ADP to ATP. So we take that phosphate, we have enough energy made from metabolism to place a phosphate onto adenosine diphosphate to make adenosine triphosphate. So I just want to try to clear that up. I know metabolism can be confusing for folks, and it was for me when I was in your all seats. So do we have any questions? Are we okay there? All right. We'll just holler if you have a question. We're about done. I know we're going over a lot. We're about done. Yeah, these muscles are quick and short and kind of to the point because there's so much information. And if I do have my students have asked me to place these on some kind of accessibility board. So I made a YouTube. So if you want this on YouTube, you can tell me to post it on my YouTube, and then you guys can go on there and rewatch it if you prefer. I'll usually put my labs up there, but if you guys want to look into it or you request it, then I can do it for you. I just kind of gotten away from doing every single one of them because I don't think people were more interested in certain spotlights than others. Okay, so when we have rigor mortis, we die. Okay, we are no longer absorbing, we're no longer inhaling oxygen. Oxygen no longer becomes the final electron carrier. And if you think, then we are no longer able to make ATP. And if you recall, if we are no longer able to make ATP, then we are no longer allowed to have the myosin head become unbound to the actin. Okay, that's how we get this stiffness, this contraction. So that's kind of explains this. So we have to have, when myosin binds, it's ADP, all right, and then when myosin unbinds, it needs ATP, all right. It needs ATP to unbound, and without ATP, it remains bound, and then we kind of go into this rigor mortis, this stiffness. And it can honestly damage the muscles over time because muscles will produce enough lactic acid where that's how you get muscle aches and pains, just through the lactic acid because of the low pH. So does anybody in the last concept here, and I think we're going to be done, does everybody know what a heart attack is? Like, give me your best idea of what a heart attack is. What's your definition of a heart attack?
Is it a blood clot in the heart?
Okay. Well, for silence purposes, I'll just kind of mention it. When there's a heart attack, yeah, Rolson, that's a lot of the truth behind a heart attack. So we have these plaques, right? We probably don't eat well. We have these coronary arteries that feed the heart, and sometimes when we have a poor diet and along with poor genetics with it, or we're diabetic, or we smoke, sometimes these lipids or cholesterol will become lodged in the coronary arteries, which are the arteries that actually help supply the heart itself, right? The heart, it's a muscle too, and it has needs, and the coronary arteries help supply this. When we have deposits in the coronary artery, we produce these plaques. This is atherosclerosis, and when we produce these plaques, the tissues underneath the plaque do not receive enough blood, and they subsequently begin to die off, right? Well, when this plaque were to become lodged and we're not no longer perfusing blood into a certain area of the heart, this kind of happens on a much larger scale, and this is what happens when you have a heart attack. Does everybody know how they diagnosed a heart attack? Like, how do they know that you're having a heart attack? There's different ways, but there's one particular way that really determines how bad your heart attack is. Yeah, chest pain's one. Chest pain's one. EKG is another. That's the sinus rhythm. That's pretty much when they look at the sinus rhythm, whether you have a STEMI versus a non-STEMI heart attack. Chest pain, left arm pain. But when they draw your blood, they order one specific test. And do we know what that is? We'll try to steer you guys in the right direction. If you don't know what it is, it's fine, but it's gonna help you remember why we talk about this. And I don't want to give the answer away too soon. Yeah, that's right. Keisha got it. Troponin. All right. So when they were to draw your blood, the doctor will order a troponin. This is the same troponin that we talked about earlier. Okay. When we have when heart troponin is so specific to the heart that this is what we measure to gauge the degree of your heart attack. Okay. When tissue dies, tissue tends to break off into the circulation. And when we have cardiac damage, troponin is the one thing that is really specific to the heart that is a good gauge of how much damage has been caused by the heart attack. This is the same troponin, if you recall, where the myosin head binds to the actin filament itself. All right. And we'll kind of next week, we're kind of going to dive into the cardiac heart. What happens when the action potential becomes a little bit changed, and why. I want to introduce it real quick. So we all kind of introduce this under the skeletal muscle. There's smooth muscle and cardiac muscle, and everybody's different. When we have cardiac tissue that contracts, it has to travel a far distance, right? When we initiate the SA node at the very top, which initiates the action potential, right? Normally it comes from our brain here, but in the heart, it has its own electrical conduction system. And it's called the SA node. It's a group of specialized cells that can fire an action potential to the whole heart, and that's what produces our lub dub. So, but it has to happen very quickly. And there's one mechanism that's a lot different than cardiac tissue versus skeletal tissue. And this is what happens when we have gap junctions in our cardiac muscle cells. And what these gap junctions do is that they really help lower the resistance of the action potential so it can travel a lot faster. Because if you think about it, we have to contract the heart from the bottom up, right? There's no valves at the bottom. We're not squishing. It's like toothpaste, right? If you want to get your toothpaste out of the tube, I guess, you probably want to press down at the bottom, right? Because all the valve is at the top, and that's where the toothpaste comes out of. Same here. So we have to make up considerable distance. We have to be fast enough to contract the heart at the same time, but slow enough to allow the blood to flow from the atrium to the ventricle. We also contract the atrium, which is at the top. So we contract the atrium, which kind of pushes the blood into the ventricle, but then we have to get all the cells ready to go and then contract at the same time to pump out the blood into the aorta or a pulmonary valve. And we can do this by gap junctions. Gap junctions help propagate they're found in the cardiomyocytes, cardiac muscle cells, and this is how they are able to contract the heart. So I don't want to go into too much depth with that. We'll probably save that for next week when we talk about smooth muscle and cardiac muscle and the different action potentials. So any questions? And I think you guys have been pretty quiet today. Thank you for your participation. I'm going to go find and being patient with me. I apologize. We're gonna wrap this up today. This is all I kind of want to talk about. I think that's enough information. And if you have any questions, feel free to lay it on me either through the chat or the microphone, and I'm going to find the attendance form real quick. 1 second. I know the cats are playing too, which I'm sure that doesn't help things. Hold on 1 second. Yeah, Keisha, I sure can. Yeah. If you're not familiar, if you're not familiar, just type in my name and like Bio 137 Spotlight Session or something like type in my last name and Bio 137. You'll see all my stuff come up. That's my channel, as people tell me. I'm kind of new to it, so it's kind of cool, I guess. Watch yourself on the internet. All right, guys. So there's the link on the form there. Fill that out. I'll hang out until you're done. And when you're done, just let me know. And so I'm not sitting here waiting on everybody when everybody's done. And then we'll catch you next time. Hey guys, are you having problems with the attendance form? Let me know if you're still out there. I'll hang around for a little bit longer in case you're working on it. Okay. All right, Rolston, thank you. We'll see you next time. All right, Darian, if you're still out there, I'll answer any questions if you're having problems with the attendance form. Sometimes students kind of step away and they don't realize that these sessions have ended. But I'll hang out a little bit longer.